Hi, and welcome to another lesson with Ms. Washington. Today we're going to talk about DNA versus RNA, talking about how two nucleic acids have specific functions in helping us code for our traits. So let's review DNA. DNA is deoxyribonucleic acid. It's a nucleic acid that carries your genetic information. It's our ultimate instruction manual. Everything that we are, everything that we will be is right there in our DNA. It codes for making proteins. It's our instructions. DNA is located in that special organelle called the nucleus. As eukaryotes, our DNA is protected by that membrane-bound organelle. Chromosomes are strands of tightly wound DNA. So they're strands of organized DNA. Sections of the DNA called genes are what actually code for different proteins. So if you remember from my last lecture, we said the chromosome was like the book. The DNA was like the chapters. And the genes are like paragraphs. Chromosomes are made up of bunches of organized DNA. And sections of that DNA are called genes. Books are organized into chapters, and sections of those chapters are called paragraphs. Not all of our genes code for DNA. Some of them don't carry information. So think about chromosomes like this. Chromosomes are DNA when it's tightly and neatly packed. It's tightly wound in a perfect ball of yarn. When the DNA is not organized, and when it's not organized into a chromosome, it's called chromatin. It's just long and stringy, and it looks like loose spaghetti. Let's refresh. The building blocks or the monomers of DNA are nucleotides. Nucleotides have three parts: a phosphate, a sugar, and a base. DNA has deoxyribose sugar, a phosphate, and one of four nitrogen bases, either adenine, A, thymine, T, cytosine, C, or guanine, G. The way I like to remember this, can you remember the way to remember the three parts of a nucleotide? Right, the phosphate is like your pool. The sugar is like your house. And the nitrogen base is like your garage. So there are four different kinds of DNA nucleotides. There's a phosphate sugar adenine, a phosphate sugar thymine, a phosphate sugar guanine, or a phosphate sugar cytosine. The only parts of a nucleotide that ever change, of a DNA nucleotide that ever change, are the nitrogen bases. The structure of DNA is double-stranded, meaning that there are two strands. The structure of the DNA is in a double helix. It's twisted together like a twisted ladder. The phosphate and the sugars always make up the backbone. The phosphates and sugars never change. So imagine you have a ladder. The sides of the ladder are the phosphate and sugars. Your paired nitrogen bases in the middle, these are like your rungs. So the paired nitrogen bases that you put your hands and feet on when you climb up the ladder, those are your rungs. Those are your nitrogen bases. So remember, our complementary base pairing rule for DNA, base pairs are adenine always pairs with thymine, A's always pair with T's, and cytosine always pairs with guanine, C's always pair with G's. So let's practice. Let's write our base pairing rule at the top. A pairs with T and vice versa. T pairs with A. That's why my arrow goes back and forth. And G pairs with C, and C pairs with G. That's why my arrow goes back and forward. So DNA is double-stranded, and you must have a complementary complementary base pair on the other strand. So let's figure out our complementary DNA strand from our template strand. So what does A pair with? Refer back to our legend. Right. A pairs with T. What does T pair with? Right, and vice versa, T pairs with A. And what does C pair with? Right, G. What are our next three complementary base pairs? Right, C, C, T. And last but not least, what are my complementary base pairs? Right, G, G, C. Good job. So the function of DNA, the main function of DNA is to code for our proteins. Each gene, each section of DNA tells us how to make one protein. So there's a gene for coding for your eye color. There's a gene for coding for your height. There's a gene for coding for your hair texture. There's a gene for coding for your dimples, or a gene coding for your hitchhiker's thumb. Each one of those genes tells the ribosome how to make a protein, or tells you how to make a protein. The sequence of those bases is where the information is actually stored. The order of those bases is different for each protein. So it's unique for each protein. That's how we get different proteins, because the order of A's, T's, G's, and C's are different for each protein. Remember, proteins determine your traits. Your traits just help you regulate growth and development. So if you need to make lactase, or if you need to make keratin or melanin, there's proteins for that. So there's a protein that's going to be made from this strand. And then there could be a protein that's made from this strand. Notice how the sequence of bases, or the order of those letters of those nitrogen bases, are different. So the kind of protein that it's coding for is probably going to be different. This is where our information is stored, in the sequence of those bases. Now, what is RNA? Well, RNA stands for ribonucleic acid. And RNA has a wide range of functions, like coding and regulating for the expressions of our genes. RNA is located in the cytoplasm and the ribosome. So unlike the DNA that has to stay in the nucleus and be protected by that nucleus, this RNA, this kind of nucleic acid, it can roam around the cell. The building blocks of RNA are very, very, very similar to the building blocks of DNA. The building blocks of RNA are nucleotides, and they contain 3 parts as well. The 3 parts of a nucleotide, of an RNA nucleotide, are the phosphate group, but the sugar is now ribose, not deoxyribose, but ribose. And one of 4 nitrogen bases. While it shares the same bases as DNA, adenine, cytosine, and guanine, uracil is the 4th base that is different than the 4th base in DNA. DNA had thymine, but RNA has uracil. So DNA has T's, and RNA has U's. So there are 4 different kinds of RNA nucleotides. There's a phosphate sugar adenine, a phosphate sugar guanine, a phosphate sugar cytosine. Those are all similar to or identical to the nucleotides of DNA, but the 4th nucleotide for RNA is a phosphate sugar uracil, not a phosphate sugar thymine like DNA. However, RNA still possesses a complementary base pairing rule. Because RNA has a wide range of functions and helps with coding, it has a complementary base pairing rule too. So in RNA, A's pair with U's and C's still pair with G's. Please note, well, nope, let's keep going. So A's pair with U's. So apples are under a tree and cars still park in a garage. There are 3 types of RNA. While there's only one kind of DNA, there are 3 types of RNA. There's mRNA or messenger RNA, tRNA or transfer RNA, and rRNA or ribosomal RNA. Let's talk about mRNA or messenger RNA first. Messenger RNA is single-stranded or one strand of RNA nucleotides. The phosphate sugar backbone is still the same. So the phosphate and the sugar make up this part of the strand. The nitrogen bases oops, I put a T. I wasn't supposed to put a T. The nitrogen bases are what make up the code of the RNA strand. Note, it's not double-stranded. Why do we call mRNA messenger RNA? Well, messenger RNA or mRNA carries the message from in the DNA from the nucleus to the ribosome during a process called protein synthesis or making proteins. We'll talk about that later on this unit. The RNA is able to carry that message that has the instructions for whatever protein needs to be made. DNA cannot leave the nucleus. However, RNA can. So RNA gets made in the nucleus and is able to pass through that port, through those nuclear pores, in order to carry the message to the ribosome. Think about your RNA, your mRNA is like your middleman. It's like playing telephone. But in this case of playing telephone, you better make sure that message is correct from start to finish. So let's practice. Let's say we're trying to make a strand of mRNA from the DNA in the nucleus. Let's practice. We're going to go ahead and write our key or our legend at the top. So when writing mRNA, when creating an mRNA strand from a template of DNA, these are the bases that match up. G still pairs with C, and vice versa. C still pairs with G. However, if you see an A in DNA, it now pairs with a U in RNA. And once we talk later on, sometimes the mRNA is going to pair up with more RNA in order to make the protein. So if there is a U, U's pair with A's. There's one more quick note I want to add. Miss Washington, DNA has T's, but mRNA doesn't have T's. So if there's a T in DNA, what does it pair with in mRNA? It must have a pair, right? Right. T's in DNA pair with A's in RNA. But if there is an A in DNA, it pairs with a what? A U. So that's why my arrow doesn't go back and forth when pairing T to A, because A's don't pair with T's in RNA. A's in DNA don't pair with T's in RNA because RNA doesn't have T's. But if there's a T in DNA, it will pair with an A in RNA. Let's do a little practice. I know it seems a little confusing. Let's do a little practice. So there's an A. There's an A in DNA. However, there are no T's in RNA. So what does A pair with in RNA? Right, U's. But look at this. There's a T in DNA. What do T's pair with in RNA? Right, A's, because RNA still has A's. And this guy, he's universal. He never changes. He never switches up on you. What do C's always pair with? Right, G's. So what are my next three complementary base pairs on my mRNA strand from my template DNA? What do G's always pair with? Refer back to our rule. Right, C, C, C. Good job. And let's do this one, the last one. Here we go. There's an A in DNA. What do A's pair with from DNA to RNA? Does RNA have T's? No, it has U's. So A's pair with U's in RNA. Uh-oh, we have two T's in DNA. What do T's pair with in RNA? Right, A's, because RNA still has A's. Good job. The other two types of RNA are transfer RNA, or tRNA, and rRNA, or ribosomal RNA. Transfer RNA transfers the proper amino acid to the ribosome by finding its match on the mRNA during a process called protein synthesis. We will talk about that later on in this unit. rRNA, or ribosomal RNA, is just an important and integral part of the ribosome. The ribosome builds proteins based off of the information from the mRNA. rRNA also helps with translation. We will talk about this later on this unit. So let's recap. Let's review. There are three major differences between DNA and RNA. Deoxyribonucleic acid and ribonucleic acid have three major differences. The first difference is their sugars. DNA has deoxyribose sugar in its nucleotides, whereas RNA has ribose sugar in its nucleotides. They differ in the kinds of nitrogen bases they have. DNA has adenine, guanine, cytosine, and thymine. So DNA has A's, T's, C's, and G's, whereas RNA has adenine, cytosine, guanine, and uracil. RNA has A's, U's, C's, and G's. Lastly, they differ in size. DNA is double-stranded and a lot larger, whereas RNA is single-stranded and smaller. Thanks for watching, and make sure you complete your comprehension questions. Have a wonderful day.